Hi guys, I'm Yogita Khandelwal and in this lesson we will deal with testes in human male reproductive system. Follow me for more lessons of biology on Unacademy and here's the link for the same. So we have already dealt with the introduction of testes in our previous lesson. Now here we will deal with covering of testes. Now testes has three covering basically. Tunica vaginalis, Tunica albuginea and Tunica vasculosa. Now Tunica albuginea is the outermost covering. It is double layered and it is a partial covering. So it does not completely cover the testis. It is only a partial covering and it is double layered. Between two layers, fluid is present which act as shock absorber. So it is very critical. Since it has two layers, so between these two layers, some fluid is present which act as shock absorber, thus protects the testis. So tunica vaginalis helps in protection of the testis. Next, we have tunica albuginea that we will deal in detail later, and the tunica vasculosa, that is the innermost layer which lines testicular lobules. We will also see what are testicular lobules. Now the tunica albuginea, as we have seen, it is the middle layer of the test which covers the testis. Now it is complete connective tissue layer. So it is totally connective tissue layer. Now it is an extensions that is known as testicular septa. So there are various testicular septa which divides testis in 250 compartments. So each testis has 250 compartments which are known as testicular lobules, and these testicular lobules are lined, as we have already seen, by tunica vasculosa. So the septa of tunica albuginea divides testes in 250 compartments. Now this is extremely important, right? Please remember it. Each lobule contains 1 to 3 seminiferous tubules, Lydig cells and immunologically competent cells. Seminiferous tubules, Lydig cells and immunologically competent cells. Extremely important. Now see, this is the TS of testis. Now see, this outermost is tunica vasculosa. Now this is the partial covering. It does not cover the testis entirely. Then this is epididymis, where it is head of epididymis. This is body of epididymis. Then this is tunica albuginea. See, this covering is tunica albuginea and here's the testicular septa. This is testicular septa and it divides testes in 250 compartments, that is known as testicular lobules. See, and in this we have 1 to 3 seminiferous tubules and the Lydig cells and immunologically competent cells. This is spermatic cord that is going into these are cremaster muscles, and this is the reti testis. Next is seminiferous tubule. As we have seen, each testicular lobule has 1 to 3 seminiferous tubules. Now these are the functional unit of testis. Remember, these are the functional unit of testis, line on its inside by two types of cells. Now two types of cells are present. First is male germ cell or spermatogonia, for undergo meiotic division leading to sperm formation. So firstly, spermatogonia forms primary spermatocyte which undergoes meiosis, forms secondary spermatocyte, spermatid, and finally the sperm. So all these structures are present at various stages in seminiferous tubules. Now these spermatogonia or spermatogenic cells are extragonadal in origin. These cells originate from extraembryonal mesoderm yolk sac membrane and enter within the fetal testis. So it is, remember, it is extragonadal and it originates from extraembryonic mesoderm yolk sac membrane. Second, we have Sertoli cells, that is also known as sustentacular cells. We will deal with these cells later. Now the region outside seminiferous tubules contains small blood vessels and interstitial cells or Lydig cells, extremely important cells, which synthesize and secrete androgens. Now the luteinizing hormones, that is LH or also known as ICSH in males, that is interstitial cell stimulating hormones. They stimulate these Lydig cells which synthesize as well as secretes androgens. Now see, this is a single seminiferous cross section of now these are the cells of Sertoli. These are the spermatogonium, spermatocyte, and these are the various sperms, and this is the lumen of seminiferous tubule. Here's the another picture showing cross section of seminiferous tubule. So this is the lumen. Here we have spermatogonia, primary spermatocytes, and there are Sertoli cells, or see these are the Sertoli cells or substanticular cells, spermatids, and outside the seminiferous tubules we have Lydig cells or interstitial cells, or we have small blood vessels, arterioles and extra, and so on. And finally, the functions of Sertoli cells. Now we have seen seminiferous tubules are lined by two cells. One is male germ cells and second is Sertoli cells or substanticular. So first function is testis blood barrier. Isolate spermatogenic cells from blood contact. So they isolate spermatogenic cells from blood contact. So why they need to be separated? Because spermatogenic cells contain protein which can generate immune response if it comes in contact with blood. So to prevent the immune response, we need testis blood barrier. They are also nurse cells because they provide nutrition to spermatogenic cells. Third, it phagocytize surplus cytoplasm of spermatogenic cells during spermatogenesis. So during the formation of sperm, they phagocytize the surplus cytoplasm so that sperm remains small in size. Then is the spermiation, very important. Now spermiation is the process which is release of sperm that is embedded in Sertoli cells into the lumen of seminiferous tubules, which helps into the movement of sperm into intertesticular sperm, that is retitestis. Intertesticular sperm conducting dung after spermiogenesis, that is retitestis. So this Sertoli cells helps in the release of sperm into the lumen of the seminiferous tubules. And lastly, in fetal stage, secretion of anti-Mullerian factor. So what is the Mullerian factor? So Mullerian factor is the factor which helps in the development of female reproductive tract like oviduct, uterus, etc. So in fetal stage, these cells secrete anti-Mullerian factor which inhibits development of Mullerian duct. So that's all for this lesson. Thanks for watching.